Welcome to BioCrack Academy. In this video, we will cover MCQ question on the microscopy with clear answers, perfect for quick revision and exam preparation. So, let's get started. The first question is, what is the primary function of a microscope? To measure the weight of specimens, to magnify and visualize small objects, to analyze chemical composition, to detect sound waves. And the correct answer is to magnify and visualize small objects. Question number 2. Which type of microscope uses visible light to illuminate specimens? Scanning electron microscope, transmission electron microscope, optical microscope, or atomic force microscope? And the correct answer is optical microscope. Third question is, what is the maximum magnification of standard optical microscope? 100x, 1000x, 1000x, or 1 lakh x? And the correct answer is 1000x. Question number 4. Which microscope provides the highest resolution? Optical microscope, transmission electron microscope, scanning electron microscope, or confocal microscope? And the correct answer is transmission electron microscope. Question number 5. What is the purpose of staining in microscopy? To kill the specimen, to improve resolutions and contrast, to reduce magnification, to measure specimen size? And the correct answer is to improve resolution and contrast. Question number 6. Which part of a compound microscope holds the objective lenses? Stage, nose piece, eyepiece, or condenser? And the correct answer is nose piece. Question number 7. What does the condenser do in a optical microscope? Magnifies the image, focuses light onto the specimen, adjusts the stage height, holds the slide? And the correct answer is focuses light onto the specimen. Question number 8. Which microscope is best for studying the surface topography of specimens? Transmission electron microscope, scanning electron microscope, phase contrast microscope, or fluorescence microscope? And the correct answer is scanning electron microscope. Question number 9. What is the resolving power of a microscope dependent on? The wavelength of light or electrons, the color of the specimen, the size of the stage, the weight of the microscope? And the correct answer is the wavelength of light or electrons. Question number 10. Which microscope is suitable for observing live cells without staining? Transmission electron microscope, phase contrast microscope, scanning electron microscope, or fluorescence microscope? And the correct answer is phase contrast microscope. Question number 11. What is the total magnification when the ocular lens is 10x and the objective lens is 40x? 50x, 400x, 4000x, or 100x? And the correct answer is 400x. Question number 12. Which scientist is credited with inventing the first compound microscope? Antony van Leeuwenhoek, Robert Hooke, Hans Janssen, or Louis Pasteur? And the correct answer is Hans Janssen. Question number 13. Which type of microscope uses a laser beam to create 3D images? Confocal microscope, scanning electron microscope, transmission electron microscope, or optical microscope? And the correct answer is confocal microscope. Question number 14. Which microscopy technique enhances contrast by using interference? Dark field microscopy, differential interference contrast microscopy, fluorescence microscopy, or confocal microscopy? And the correct answer is differential interference contrast microscopy. Question number 15. Which microscope uses a beam of electrons to study internal cell structures? Scanning electron microscope, transmission electron microscope, phase contrast microscope, or fluorescence microscope? And the correct answer is transmission electron microscope. Question number 16. What is the function of the diaphragm in a microscope? To magnify the image, to regulate light intensity, to hold the slide, to focus the lenses? And the correct answer is to regulate light intensity. Question number 17. Which microscopy technique uses fluorescent dyes to label specific molecules? Confocal microscopy, fluorescence microscopy, phase contrast microscopy, or dark field microscopy? And the correct answer is fluorescence microscopy. Question number 18. What is the minimum resolving power of the human eye? 100 nm, 100 micrometer, 1 millimeter, 10 centimeter? And the correct answer is 100 micrometer. Question number 19. Which microscope is best for studying the 3D surface of a specimen? Transmission electron microscope, scanning electron microscope, optical microscope, or phase contrast microscope? And the correct answer is scanning electron microscope. The next question is, what limits the resolutions of a light microscope? The size of eyepiece, the wavelength of visible light, the weight of the stage, the number of objective lenses? And the correct answer is the wavelength of visible light. Question number 21. What is the numerical aperture of a microscope lens related to? The magnification power, the light gathering ability, the weight of the lens, the color of the specimen? And the correct answer is the light gathering ability. Question number 22. Who discovered cells using microscope? Antony van Leeuwenhoek, Robert Hooke, Matthias Selden, Theodor Swan? And the correct answer is Robert Hooke. Question number 23. What is the primary advantage of electron microscopes over the optical microscopes? Lower cost, higher magnification and resolution, ability to view live specimen, or simpler operation? And the correct answer is higher magnification and resolution. Question number 24. Which component of a microscope adjusts the focus of the image? Stage controls, fine and coarse adjustment knobs, diaphragm, or nosepiece? And the correct answer is fine and coarse adjustment knobs. Question number 25. What type of microscope is used to study magnetic domains in materials? Scanning electron microscope, transmission electron microscope, Lorentz microscope, or confocal microscope? And the correct answer is Lorentz microscope. Question number 26. What is the role of immersion oil in microscopy? To clean the lenses, to increase numerical aperture and resolution, to stain the specimen, to reduce light intensity? And the correct answer is to increase numerical aperture and resolution. Question number 27. Which microscope technique uses polarized light to study birefringent material? Polarizing microscope, fluorescence microscope, phase contrast microscope, or confocal microscope? And the correct answer is polarizing microscope. Question number 28. What is the typical thickness of a specimen for transmission electron microscope? 1 to 2 micrometer, 50 to 100 nanometer, 10 to 20 millimeter, 1 to 2 centimeter? And the correct answer is 50 to 100 nanometer. Question number 29. Which part of the microscope holds the slide in place? Stage clips, condenser, eyepiece tube, objective lens? And the correct answer is stage clips. Question number 30. What is the main limitation of scanning electron microscopy? Cannot view live specimens, low magnification, requires no preparation, uses visible light? And the correct answer is cannot view live specimens. Thank you. If you like the video, please share, comment and subscribe. Thank you again.